In this video, we will be going over the mechanisms of neurotransmitter clearance from the synaptic cleft. And the way I broke down this video is we will go over the mechanism, a description of the mechanism, some examples, and their clinical relevance. So getting started, for the first mechanism, we have reuptake, which is when neurotransmitters are taken back into the presynaptic neuron via transporter proteins. Some examples include serotonin, SERT, serotonin transporter, dopamine, DAT, dopamine transporter, and norepinephrine, NET, norepinephrine transporter. And their clinical relevance includes SSRIs, which are selective serotonin reuptake inhibitors, which block SERT to treat depression and anxiety. And then we also have SNRIs, which are serotonin norepinephrine reuptake inhibitors, which block NET and SERT. The second mechanism involves enzymatic degradation, which is when neurotransmitters are broken down by enzymes in the synaptic cleft. And this includes acetylcholine, which is broken down by acetylcholinesterase into acetate and choline. And then dopamine and norepinephrine, which are broken down by monoamine oxidase, abbreviated MAO, and catechol-O methyltransferase, abbreviated COMT. And the clinical relevance here is that MAO inhibitors, also known as MAOIs, block the breakdown of dopamine and norepinephrine to treat depression. And then also acetylcholinesterase inhibitors are used to treat Alzheimer's disease. The third mechanism, and perhaps the most common one, is diffusion, which is when neurotransmitters diffuse away from the synaptic cleft into the extracellular fluid. And this is a general mechanism for all neurotransmitters. They are less efficient clearance mechanisms and are often supplemented by reuptake and enzymatic degradation. The fourth mechanism is uptake by glial cells, which is when neurotransmitters are taken up by surrounding glial cells, for example, astrocytes. And an example of this is glutamate, which is taken up by astrocytes via excitatory amino acid transporters, also known as EAATs. And their dysregulation can contribute to excitotoxicity and neurodegenerative diseases. And lastly, we have endocytosis, which is a rare mechanism where the postsynaptic neuron may internalize neurotransmitter receptor complexes. And it's only found in specific instances with neuropeptides, and it's a much less common mechanism, more relevant for certain signaling peptides and proteins.